Join with computer audio. Courtney. Whoops. Got 5 people all coming in now, Claire. Hey, that's Anna. Hi, Anna. Can you guys hear me? Can you all hear me? Can somebody nod yes? No? Can any of you hear me yet? All right, so we got to do something. No, we got to turn on the audio somewhere here.
I can hear you.
Oh,
This
okay.
is Courtney.
All right, thanks, Courtney. So this is my little great niece, Claire. She's coming in to say hi and wish you all good luck on your final exams. So I got, I don't think I have COVID again, but I have a red throat, don't I? Mm-hmm. And so Miss Claire is an Allen Park student. And so thanks for coming tonight. I have a whole house full of people here for Thanksgiving. And look, this way we got Kiara, everybody here. Kiara, yay. She says she's feeling okay after her surgery. So good job there. All right, let him wave bye. Okay, that's her hand waving bye. All right, make sure you close the door on the way out. So tonight we're going to do vision, okay, instead of general senses, just because it's pretty easy to do over the internet here, okay? So we'll go through vision. And our basic objectives for learning the vision is we're going to first learn the structures of the eye, so both the structures of the eye itself and the accessory structures like the lacrimal glands. And then we'll go through the eye muscles that move the eye. And then we'll look at some of the sort of more internal muscles, things that like control the pupillary diameter. We'll go through the tunics of the eye or the layers of the eye. And then we'll discuss what are the cavities sort of in the chambers, sort of the anterior chamber, posterior chamber in the of the eye. Then we'll talk about how the light is processed through the eye, which is really important. And that's a lot of the physiology and focusing the light onto the retina. And then, of course, we have to look at the photoreceptive cells, the rods and the cones. You've probably learned those a little bit before. And then we'll go through disorders of the eye. I have disorders of the eye and ear here, but we're not doing hearing tonight. We're just doing vision. So, all right. All right, so when we're starting with our eye, at the outside of the eye, we have our eyelashes and our eyebrows. The hair, which their basic function, as you could probably guess, is going to be to trap dirt and dust and help keep it out of the eye. And then we have our eyelids. And our eyelids, when you fold them up and look back, you can see that they're contiguous with the conjunctiva that covers over the white part or sclera of the eye. And the conjunctiva is a mucous membrane, so it's wet epithelium. And we call the area actually in between the two parts of the eye the palpebral fissure, okay? Now, the angle of the eye right over here is called the canthus, okay, where the upper and lower lids join. And the eye ball itself will have 3 distinct layers: the outer tunic, a middle vascular layer, and then the innermost layer is going to be a nervous cell layer, and that's where all the photoreceptors will be. All right, so here you can see the eyelashes and the eyebrows, and they serve basically the protective function. They'll keep out all that foreign debris and also capture your perspiration so it's not dripping in your eye. If ever you've worked out really hard and you had some of your really salty perspiration get into your eye, it kind of stings and burns your eye. I'm sure some of you have had that happen before. The eyelid itself is going to, as you blink, it's going to help spread the tears all over across the film of the eye. And it also helps keep your eyes from drying out when you're asleep. And of course, when people are in the hospital, pretty often we have to tape their eyes shut. We tape them shut when they're in the OR to help keep the eyes moist. And sometimes on the floor, you'll learn how to tape people's eyes shut if they have, some problem that's keeping their eyes open. Your blink reflex will help you blink out when you have a foreign body or like an interned eyelash or something like that in your eye. You're going to blink it so that it passes over to the corner of your eye. And the words palpebral and blepharal both mean having to do with the eyelid. So if you have blepharitis, you're having an inflammation of your eyelid. We also see that in certain ethnicities, like in Asian folks, you have a little extra skin fold over here, and that's called an epicanthal fold, okay? Doesn't, affect your eyesight or anything like that. It's just found to varying degrees in different populations, all right? So, outside of our eye, we have the lacrimal apparatus. You have your lacrimal gland that's always going to be out at the lateral part of the eye, and it's going to produce the tears that will then, as you blink, move across the eye in toward the medial portion of the eye, which is going to be at the canthus. And in each canthus, you're going to see that you have this kind of swelling of tissue called the lacrimal caruncle. And right around it, you're going to have little puncta or little holes that will lead in to the lacrimal canals that then lead to the lacrimal sac inside of your nasal canal and down the nasolacrimal duct. And this is why when you cry and tear up, you also swell up in your nose and everything sort of starts extruding from both your eyes and your nose. We have 6 external eye muscles that move our eyes in all of the directions. So the superior straight muscle going back that moves our eyes to look upward is going to be our superior rectus. Inferior rectus down below, we will have on each side of the eye, we'll have lateral recti and we'll have medial recti. And then we'll have some that come at a diagonal, a superior oblique and an inferior oblique. And the muscle that controls the elevation of our eyelids is the levator, like elevator, open it up. Palpebrae opens up the distance between our two eyelids. All right, so one of the things that always drives students crazy is learning what each of these eye muscles do, which isn't so hard in and of itself, except for the oblique muscles. So as you can imagine, the superior rectus moves the eye up, the inferior rectus moves the eye down, the lateral rectus will move the eye to the lateral side, the medial rectus will move the eye over towards your nose. And so when I look over to my left, I'm going to be using my lateral rectus on my left eye, but the medial rectus on my right eye to move it that way. And the opposite when I move my eyes to the right lateral rectus, left medial rectus. So we have movement of our eyes, of our vision, field of vision, okay? Where it gets hard is with our inferior and superior oblique. And this doesn't intuitively make sense. And sometimes I recommend if you have a ball, like a little tennis ball, kind of play around with it by drawing a little pupil in the center and then putting a piece of tape. We sometimes do this in lab and then kind of contract or shorten it, and you can see how it moves. All right, so let's look at this. So your superior oblique comes across this way and goes through this little trochlea. All right, it's got a little pulley system. And when we shorten or pull on this, it's going to pull the eyeball this way. So the eye goes down and kind of inward like, okay? It's kind of moving in this direction. When we have the inferior rectus and we shorten and we pull on it this way, we're going to rotate the eyeball around like this, and it kind of moves upward, all right? Now, you also need to know the innervation of each of these eye muscles. So all of them are innervated by the oculomotor nerve, cranial nerve number 3, except for superior oblique, cranial nerve number 4, the trochlear nerve. That's the only thing it does. All right. And number 6, the abducens or abducting nerve moves the lateral rectus. So moves your eye outward to the side. Okay. The mnemonic, which may or may not be helpful to you, is like memorizing a chemical formula. So it's LR6SO4, lateral rectus, cranial nerve 6, superior oblique, cranial nerve 4. Everything else is cranial nerve 3. Okay. Hopefully that helps you. If not, you can come up with your own mnemonic or try to look up another one, but that's kind of the classic one that everybody uses. All right. So here from the side, you can see all the different eye muscles. Sorry, my mouse is a little trigger happy. Superior rectus up here, moving the eyeball upward. Inferior rectus will pull the eye downward and straight. Lateral rectus over here. This is mislabeled. Okay, I can see that. Oh, you know what? I'm sorry. We're looking down the eye from above. So this is superior rectus. Okay, I'm sorry. That's why I was confused by this. Superior rectus, this is medial rectus. This is moving toward the nose over here. This is moving outward toward the zygomatic arch over here. Here's lateral rectus, superior, so pulls this back, moves the eye upward. And then you can see the superior oblique going through the trochlea, the little trolley or pulley notch, all right? And this one that's cut away is the one that goes to the eyelid, the levator palpebrae. All right, here you can see it from the side view, okay? So this is sort of the upper forehead. This is down on the lower cheek. Here's your superior rectus. Here's your superior oblique. Coming over here, your inferior oblique, inferior rectus pulling straight back. And here is levator palpebrae. And you can see the back part of the inferior rectus down here. All right. Now, those were all the external ocular muscles. We also have internal or intrinsic eye muscles. And the main thing here is going to be muscles that will control how open our pupil is. Okay, so your iris, even though you think of it as the color and the pigment, it's basically modified muscle tissue right here. And the iris opens and closes based on how much light is coming in. It's a diaphragm, okay, or a sphincter, okay? Next to the iris, you'll see out here is what's called the ciliary body. Okay, so you have all these circular muscle fibers. And I'll go through this because it's not entirely intuitive. Attached to the ciliary body are these zonula fibers right here, sometimes called the suspensory ligaments as well as the zonular fibers. And when this muscle is relaxed, okay, these fibers here are tight and it pulls and flattens out the lens, all right? When this muscle constricts, the muscle actually kind of bulges inward and then these zonular fibers actually kind of loosen up and that enables the lens to kind of fluff up and get thicker. And this will be part of accommodation. And we'll go what's called accommodation as your lens will adjust to looking at things that are up close to you or far away. All right. So contraction of this muscle allows these fibers here to go slack. So remember, when the muscle contracts, the fibers go slack, the lens rounds up. When the muscle ends up relaxing, the fibers pull and tighten. The muscle kind of pulls away as it relaxes. This increases the tension on these fibers and it flattens out the lens. That's what happens when you focus on a distant object. So we'll go through this again, okay, because it is not intuitive. All right. So here we see that lens accommodation, adjusting your eye as you go from near to far vision. All right. And you can see here, focusing on the light. When you're looking at a distant focus, something far away, we're not even really seeing it. All this light is coming through and it has to focus on the retina. We have relaxed ciliary muscles. When the ciliary muscle is relaxed, the fibers are tight, all right? And we flatten out the lens. Here, when we're looking at something that's close up, okay, near vision, like you're reading a book or looking at your tablet, the ciliary muscle is contracted, and you see when it's contracted, all these zonular fibers go slack, and the lens is able to round up, and you can focus, you adjust your focus onto the close object. And here you can kind of see the difference in the pupil when it's tightened, okay? We're tightening and constricting right here, all right? And again, here you see it relaxing, all right? Focusing on the close, on the near object. All right, looking at that lacrimal apparatus again, okay, we're going to make in our lacrimal glands all of our tears. Our tears are antibiotic, so we can take care of all the microbes that might land on our eyes. So part of that is you having in there an enzyme called lysozyme, the antibacterial enzyme. Remember, you have lysosomal enzymes in your phagosomes, okay? And in the tears, it's going to make all the tears, they're going to come on and form a lacrimal lake across your whole eye. Your eyelids will flick and move the tears around, and they will drain out through the puncta, or the little holes at the canthus or the angle of your eye. From that canthus puncta, the little holes, they're going to drain down into the lacrimal canaliculi, into the lacrimal sac that's right at the bridge of your nose, and travel down the nasolacrimal duct, all right? And that's where you discharge it out of your nasal meatus. All right, so the three layers of the eye, or three tunics. We have the fibrous layer on the outside. That's largely going to be our sclera, which is the back 5/6 of the eye, and the cornea, which is the front 1/6 that's over the lens. The middle layer is going to be called our vascular layer, or we call it the uvea, and that's going to contain the iris, the colored part of your eye, that's the muscular diaphragm, the ciliary body, that muscular layer that also has some epithelial cells on it that will make your aqueous humor, and then the choroid, or the real vasculature. Whoops. The innermost layer where the light strikes is the neural layer, okay? And that is your retina. That's where we're going to have the rods and the cones, the photoreceptors. All right, so when we look at the eyeball, which is also technically called the globe, we have three chambers, three separate chambers that will sit into two different cavities. You have an anterior cavity, which is all in front of the lens of the eyes, and that's what fills up with watery aqueous humor, okay? There will be an anterior chamber part of this, which is behind the cornea, but in front of the iris, and a posterior chamber that is behind the iris, but still in front of the lens. And then we have a second cavity called the posterior cavity, which is all behind the lens, and that's filled up with a more gel-like substance called the vitreous humor. And that's what gives the eyeball more its shape and kind of sustains that. All right, so two chambers, posterior and anterior, and the anterior, two cavities, anterior cavity and posterior cavity. And the anterior cavity is divided into an anterior chamber and a posterior chamber. Like I said, always just to make it a little bit more confusing for all of us to learn it. All right, so in these chambers, we'll go through the parts of the eye, and we'll talk about how the aqueous humor is made and circulates. So we'll come to see how the lens of the eye because it needs to be transparent and we need to see through it. We need to get all the light that comes into our eye, getting it back to the retina. Our lens cells are very transparent. So we'll end up seeing that our lens is made of these crystalline proteins, and basically all of the organelles are extruded from the inside of the cell. So you just have a shell of a cell left, all right? And there's no blood vessels because we don't want light having to pass through blood vessels. So to keep everything as transparent as possible and to still feed the lens cells, the lens is instead bathed in aqueous humor. Okay, that aqueous humor is made by epithelial cells that cover that ciliary body. Ciliary body, mostly muscular, has all those zonular fibers attaching to it, but there are epithelial cells that produce the aqueous humor. That aqueous humor will then float over the lens. And let's just kind of look at the picture as I talk us through this. All right, so here we have our ciliary body, and it's covered in some epithelial cells. Here is our posterior chamber, which is behind the iris, and this is our anterior chamber in front of the iris. Let's just go over all these parts. Here's the cornea, which is the 1/6 of the outer tunic, and that's transparent also. Everything going through here has to be transparent for the light to go all the way back to the cornea, to the retina. Okay, so your cornea, 1/6 of the outer covering, transparent, and it meets up here or is continuous with the sclera, the white part of the eye that's white all the way to the back. Underneath the cornea, but in front of the iris, you'll see there's what's called the anterior chamber. At the iris, the diaphragm that closes with all these muscles, all right, is in the colored part, it has the pigments. This is going to separate the anterior chamber of the anterior cavity from the posterior chamber of the anterior cavity, which is right here between the lens and the iris. Back behind the lens, all this part is filled up with gel, which is called vitreous humor, and this is the posterior cavity. This ciliary body, that's the muscular part that has these zonular fibers that pull on the lens and change the shape of the lens, also has a covering of epithelial cells that make the aqueous humor. That aqueous humor is going to circulate from behind here in the posterior chamber and bathe over through the anterior chamber. It's going to supply the lens of the eye with all the nutrients, hormones, anything that the eye needs. And it's also going to take away any waste produced by the lens epithelial cells. And it comes over here to what's called this little trabecular network that surrounds the iris. This trabecular meshwork also leads into what's ultimately called Schlemm's canal. And this is how the aqueous humor that's made back here at the ciliary body circulates from posterior chamber to anterior chamber, ends up leaving through Schlemm's canal and takes through the episcleral veins all the waste material away. We'll talk about glaucoma later on, and when these trabecular meshwork in the Schlemm's canal, we're not quite sure exactly what all happens to it in the process of glaucoma, but it has dysfunction and does not remove all that aqueous humor, and so the pressure builds up inside the eye. That's called open angle glaucoma. The other type is called closed angle glaucoma, and that's when this lens and iris kind of moves forward into the anterior chamber and literally blocks the canal. That's closed angle glaucoma. That's a small part of glaucoma, and that's a medical emergency. You can go blind rapidly. Open angle glaucoma, where the trabecular and Schlemm's canal doesn't work so well, is kind of a slow chronic process, okay? All right, so remember, confusion arises between what's the anterior segment and what's the anterior chamber. So the whole anterior segment is filled with aqueous humor, okay? The whole anterior or posterior cavity is filled with vitreous humor, okay, the gel. In the anterior segment, it's comprised of the two parts of the anterior chamber and the posterior chamber. The posterior segment or posterior cavity itself is just the whole big back of the globe that's filled up with the gel of the vitreous humor. And I know it's just getting down that terminology. All right, so we've talked a little bit about the outer coats of the eye, and now we're moving into the vascular layer of the eye. The vascular layer is the middle coat, all right? And this is where we're going to have the blood vessels, and we'll also have a large amount of melanin. The reason melanin is in here is it kind of captures any light rays that kind of scatter around in the vitreous humor, okay, and absorbs it so it doesn't keep reflecting backward and blurring our vision. The vascular layer, of course, is important, as the vascular layer is everywhere, because it's going to supply us with the blood and the lymph that will carry nutrition to the eye and waste away from the eye. It also helps to regulate the amount of light that enters the eye. And you'll see how the vascular layer will secrete and reabsorb the aqueous humor. We just talked about it a little bit, circulating around the posterior and anterior chamber, comes in through those episcleral veins, through the canal of Schlemm. And it helps to control the shape of the lens when we're focusing on near versus distant objects. So we have three components of our vascular layer. I really apologize for why my mouse is so crazy. I really don't know. The uvea, the iris, the uvea, which is made of the iris, the colored part of the eye, the ciliary body that we talked about so much for controlling the shape of the lens, and the choroid, which we haven't talked about. Now, I think I missed a slide. No, I did not. All right, so now we'll talk about the choroid. The choroid is going to be a vascular layer that's between the retina, the neural layer where the rods and cones are, and the sclera, the back white part of the eye that, you know, is the outer covering or supportive covering. And the choroid is going to have all the vascular tissue that gives nutrition to the back of the eye. And like I said, this layer is going to have a lot of melanin. It's going to absorb all the stray light rays so they aren't kind of bouncing around back there and giving us glare, okay? The iris that we all see as the colored diaphragm changes size, you know, makes our pupils either big or small, will control basically the amount of light that reaches the retina and give us the color to our eye, which, you know, we respond to socially. The ciliary muscle is going to control the lens. Okay, we've already gone through, but we'll keep beating it through our heads because repetition helps us. It's going to contract, and then we get slack on those zonular fibers, the suspensory ligaments, and the lens can puff up or round out. That gives us increased focusing power. All right, so when we want to look at something near. And when the ciliary body muscles relax, it pulls tighter on those lens fibers and the lens flattens. And we have less focusing power. So when we want to look at something that's farther away. We refer to this changing from near to far and near to far vision as accommodation. Accommodation, you can see, is the shifting shape of the lens to change our focus from near to far. And as you know, as you get older, your ability to shift your focusing is a little bit more challenging, okay? Remember always that the ciliary epithelium that covers that muscular layer is what will make the aqueous humor. I think my kitty cat's going to come over here and try to visit us in a second. I can see her jumping around out of the corner of my eye. All right. So the iris, the colored part of the eye, contains muscles. That's what allows it to be a sphincter to change how much light enters the eye. When it dilates, it makes the pupils bigger. Okay. We call that mydriasis, a big dilated pupil like what I have probably right now because it's relatively dark in this room. But if I get closer to my light source, of course, I will then have miosis, which is it constricts the pupil, the pupillary muscles, so less light can enter. All right. And we call that miosis. And you'll often see certain drugs that we take can affect this. People who are chronically on opioids always have miotic pupils. It's one of the things that we kind of look to assess, addicted disorders. And if people, come into your office all the time and they, their eyes are always constricted, even like when you start flashing, light around and removing it away and their eyes should dilate. If they don't dilate much, you can be pretty suspicious for the fact that they're on opioids. Okay, that's one of the ways that we use to, like in the ER, when people are seeking drugs, trying to assess, whether this is really they're coming in unusually for pain or, whether there's somebody who's chronically on opioids. So you can kind of look at stuff like that. Of course, somebody who's been chronically on opioids and they lower the dose or they're coming down from being on opioids, one of the things that happens is their pupil gets dilated, okay? Oops. All right. So here you see bright light, pupillary muscles are circular and they constrict, okay, reducing the sphincter size. Here's normal light. And here we would see in dim light, the constrictor opens up to let more muscle coming into the eye. We always are trying to control or coordinate what's going on with our ANS, with our muscles. So doesn't always necessarily make the most sense to us, but parasympathetic in bright light, okay, is going to be under control of this circular pupillary sphincter muscles to constrict. And it's a sympathetic reaction to looking in dim light. One person shared with me their mnemonic is they found that looking at themselves in dim light was a more sympathetic view or more positive view in their pictures than when the bright light is on you and you show every lump and bump. So that maybe that's a little mnemonic that can help you with the pupillary constrictor muscles. All right, here's another slide to show you. Again, this is called mydriasis. Mydriasis is pupillary dilation. This is miosis, pupillary sphincter. Anybody remember just from the previous slide, which one is sympathetic and which one is parasympathetic? So sympathetic in dim light, okay? Big pupils, sympathetic, all right? This is parasympathetic. All right. Now let's look at the retina. I just want to make sure that I'm not skipping over slides because it has a tendency to do this with this trigger happy mouse. Okay, so we've gone from the outer layer, which is going to be the white of the eye, the back, the sclera, the 5/6, the front 1/6, that's the cornea. Then we did the whole inner middle layer, which is vascular, the choroid at the back of the eye, the ciliary muscle and the iris, okay, up at the front of the eye. And now we're doing the very inner layer of the eye, which is going to be our retina, okay? And our retina is also known as the neural layer. And it has two basic layers to it, an outer layer called the pigmented retina and an inner layer called the sensory retina. The outer layer is going to be the nourishing layer. It's attached to the choroid, that vascular layer underneath, all right? The inner layer is where we have our photoreceptors, and our photoreceptors are going to be the rods and the cones. So let's look at it here. All right, the surface of the retina, this is where light is going to be coming in through the eye right here, passing through the cornea, into the lens, through the pupil, the iris right here, through the lens here, and it's going to strike at the back of the retina right here. So this is going to be the section we take of the retina right here, and look at the surface of the retina. And here we're going to have some ganglion cells, bipolar cells, and this is what the light is really striking, which is our photoreceptor cells of cones and rods. Okay, the cones kind of in this reddish color, the rods being purple. And here's that nourishing layer of the pigmented retina, which is attached to the choroid, would be underneath. So the deepest cells are the photoreceptor cells. That's like our general receptors, like our Pacinian corpuscles and stuff that we would have for sensing feeling, all right, out in the environment. This layer of bipolar cells that connect and get information from the photoreceptors, they only connect one-on-one to each cone, because as you will find out, the cones are the ones for our sharp, clear vision. But you can see a bipolar cell will make connections with many rods. Rods are more for generalized vision and vision in dim light. All right, there will be some horizontal cells which will kind of integrate some of our information, and then ultimately the ganglion cells, which are going to perforate through the sclera and emerge to form our optic nerve or cranial nerve 2. This is what's going to take all this information from the light striking our retina and come back to give us our vision and take it to our brain. Thank you, Claire. All right, now we'll find on our retina, we have one area that's what we call our blind spot. And that's where the axons of all these ganglion cells will unite to form the optic nerve. And as that optic nerve leaves the eyeball, there's no photoreceptors there. So that's literally the blind spot, because there's nothing for light to strike and create a message. All right, so as you can see, here's this whole layer right here. We have our pigmented retina, the nourishing cells that are in contact with this would be the choroid layer. And here you see the sclera, the white layer at the back of the eye. Here's all those bipolar cells in connection with multiple rods, but one-on-one relationship with a cone. And here you have the ganglion cells and their axons all unite, and you see how they unite to form the optic nerve right here. And remember how you see the optic nerve emerging from the brain, right? This area where all these ganglion cells form the optic nerve has this little round area called the optic disc. As well as the nerve leaving, we have the main central retinal artery and vein enter and exit with that, okay? And so there's no photoreceptors right here, no rods, no cones. And so there's no vision in this area. And you've probably literally tested for your blind spot before. If we were in lab, we would test for it on each of us, but you'll see how we do that, okay? You'll see the little test, and you can download it at home, and you can play with yourself. You can play with your kids to test for your blind spot. It's kind of fun to do. All right, so looking at these photoreceptors that we see here, let's look at them up close. All right, so we have our cones. Our cones are what are going to give us our color vision, all right? They give, because they have a one-on-one relationship with the bipolar cells, they're going to give us the sharpest vision that we have. And to function, they require bright light. we'll see that they're densely clustered in one particular area of the retina that we call the macula densa. And in the center of the macula, we call the very center of that the fovea. That's the highest density of cones there is, where we have our clearest vision. And we'll talk about a little bit how we have cones specifically that innervate or send a message to the brain for the color blue. All of our primary colors, the color green are M cones and the color red are L cones. And, you know, just like when you mix your primary colors together, you know, you can mix a little bit of you know, green, of blue and red to get a more purplish color, things like that. All right. Your rods, your rods don't discriminate between colors. Your rods are going to be monochrome vision, a black and white world. And this is what allows you to see in dim light. And that's why when it's in the middle of the night and it's dark and you're getting up and you can see your way in the darkness to find your way to the bathroom, but you're not really seeing the world in color, in green light. These are most sensitive in what we call the green range of light, around 500 nanometers. In the pigment that the dim light, the little amount that strikes in a pigment called rhodopsin. And we'll see how that has to split, okay? But many, many rods will converge onto onto one single bipolar cell, okay? So the vision is not as sharp. All right, so our optic nerve, as we saw, is going to be formed by all of those ganglion cells merging to form the optic nerve. And they do so at the optic disc where there are no rods or cones, okay? And we'll see how that's a circular region that's just medial or towards your nose from that high density area of rods and cones that's called the fovea, all right? The origin of the optic nerve at the optic disc is your blind spot, right? And all of your retinal vessels also exit and enter there, the retinal artery and retinal vein. Okay, so when we look into the eye with our ophthalmoscope, we look through the light going through the cornea, through the pupil, like right around the iris, and going past the lens. It goes through the lens and then strikes on the retina. And this is what we see. Okay, so over here we see the artery and the vein, okay, emerging. This is the optic disc. This is where there's no photoreceptors. This is on the nasal side from the fovea, where we have the highest density of photoreceptors, rods and cones. And you see there's no vasculature here, nothing to interfere with the vision, okay? So if this is the nasal side, this is the lateral side, I can look at this and say that this is a left eye. You don't need to know that necessarily, but you know, you can play around with your anatomy and see if you can figure these things out. Ophthalmoscopy, can I talk? Ophthalmoscopy, I can't talk, is also called fundoscopy, and that's what usually people will say. So that's when you use the ophthalmoscope to look at the retina and get that vision of the optic disc and the fovea. The fovea, the larger area right here, is called the macula. The fovea is just the center of the macula, okay? All right, and it magnifies the vision back here, all right? And you can do this, you know, most of you have had this done. We can take pictures here, okay? And you can see the vessels, the blood vessels emerging at the nasal side right here. Here, another blood vessels, okay? You can see the fovea in the center right here. The macula is kind of outlined right here, okay? That sort of fundal area. There's the macula, and here's the fovea, kind of more the red region in the center, okay? Here's the optic disc again. Here's the macula. There's the fovea in the center. Here's the optic disc over here. So this just gives you some different views of what ophthalmoscopy, fundoscopy, let's say that, okay, is. Remember your cavities in your humors, cavity, anterior cavity, everything in front of the lens, right? Posterior cavity, all right, and back. Our anterior cavity is divided into the two parts, the anterior chamber and the posterior chamber. The whole anterior cavity produces the aqueous humor. It's produced at the ciliary body, which is in the posterior chamber. All right, and it passes from the posterior chamber, circulates around through the iris and goes into the anterior chamber. The vitreous humor, also transparent, more gel-like, located in the posterior chamber of the eye, and it helps maintain that globe shape of the eye. All right, so once again, aqueous humor made by the epithelial cells out here on the ciliary body. Again, these are the zonular fibers that when they're taut, which they look here, kind of flattens the lens. And when it bulges inward or constricts, it kind of slackens on these zonular fibers and the lens rounds up. Anterior chamber, posterior chamber of the anterior cavity. Posterior cavity filled with vitreous humor. You have your layer back here that is the retinal layer in yellow. Okay, that's the neural layer. That's where you have your rods and cones. This middle layer, okay, is called the vascular layer, contains your iris, your ciliary body, and what's called the uvea and choroid back here. And then when you have your outer tunic or layer of the eye, which is the anterior, most anterior 1/6, which is clear, which is the cornea. And then you have the white of the eye, that's the sclera. Okay, remember your ciliary body is muscular, okay, and that will help pull back on those zonular fibers. Your pupil, okay, in the iris here, you have your pupillary muscle, which can either constrict or it can open up or dilate, all right? And when the aqueous humor circulates, as it's made back here on the epithelia covering the ciliary body, it circulates from the posterior chamber, travels through the pupil out to the anterior chamber, and it will be absorbed here at the trabeculae and canal of Schlemm, all right? Now, to create our images on our retina, we have 4 processes. We're going to refract some of the light rays. We're going to have our lens changing shape to help focus or accommodate to focus our object, whether it's distant or near. We're also going to have to constrict our pupil to control the amount of light that's entering our eyes. And we'll have convergence of the eyes. So let's go through all this. All right, so refraction of our light waves will involve the light waves changing direction as they pass from one medium, like air, versus passing through, say, the tear layer or passing through the cellular layer of the cornea and then later the lens. Refraction is going to be a bending of the light rays, okay? All right, so the things that bend the light rays will be things like the cornea, the aqueous humor, the fluid there, the lens itself, and even the gel-like vitreous body. All these work together to focus the light rays on an object approximately 20 feet away. So when we say you have 20/20 vision, you can see at 20 feet away what a normal eye sees at 20 feet away. All right, we'll go through that in a little bit, the Snellen chart, but you're already probably keying on the number 20 because you know about 20/20. We all can have refractive errors. Some of us, like myself, can have myopia. Myopia means you're nearsighted. You can see close-up objects very closely, but your far away objects are blurry. In hyperopia or hypermetropia, one is considered to be farsighted. So you can focus very easily on distant objects, but clear objects, near objects are not so clear, they're blurry. So here, when light hits a surface like a mirror, it's reflected away. In refraction, traveling through a lens, the light is bent, okay? And we can vary the thickness of the lens to change the refraction. And that's basically what we do with external spectacle lenses. All right, so here you can see if everything was reflected coming into the eye, we wouldn't have any light waves coming in and stimulating our photoreceptors. So that's not very helpful. Refraction through the various layers, cornea, the watery layer, the aqueous humor, the lens epithelial cells will bend and refract the light to focus it on different parts of the retina. We could also have absorption and re-radiation, okay? But you could see how this would be harder to control, and diffraction, bending the light literally away from the eye surface, not as helpful. So light refraction is what we're concerned with, what our cornea and our lens will do. And the goal is to bend the light so that the rays coming in will intersect on a singular point on our retina to give us a clear focus point for vision. And we call it the focal distance, which is the distance between the center of the lens and the focal point of the object. So we'll look at some of this. Picture says 1,000 words on this. All right, so here we have our lens and the light rays coming in, and you see how they're all refracting because we have different bends and shapes at each point in the lens refracting to converge all of these light rays on the object, which should be back here on the retina. This distance from the lens center point to the object is the focal distance. All right, here you see you have a close object. You have your lens kind of rounded up, and here you have the focal point. Now, as we move the object even closer to the lens, it rounds up even more and brings us to a shorter focal point. Remember, not intuitive that our lens is going to round up when the ciliary muscle kind of bulges or the contraction actually moves the ciliary muscle a little bit more anterior and bulging inward. Normally, we think of when the muscle contracting that it's going to pull back on these fibers. Okay, so not entirely intuitive. There's a really good YouTube video. I think it's from the Ophthalmology Society that goes through all this. Okay, so you can see this is where the lens is rounded up. And when the lens rounds up, it actually rounds up a little bit more on this posterior side than on the anterior side. It's not a complete oval kind of shape that you always see. And we call that rounding up of the lens for near vision, or changing from distant vision to near vision, or vice versa. We call it accommodation. Okay, so the lens is going to change shape based on the pull of those zonal fibers or suspensory ligaments. Okay, this is going to help focus that visual image so that it strikes right on the densest area of the photoreceptors, which would be the fovea on the macula densa. The lens cells themselves are made from lens, what we call lens fibers. These are lens epithelial cells from which all the organelles, nuclei, everything's been extruded. And what's left is a kind of shell of a cell that has these crystalline or transparent proteins. Okay, so that's what gives it its refraction ability. And of course, the change of the shape of the lens is based on the tug and pull of those zonal fibers. Contraction equals bulging, rounded up lens. relaxation equals pulled taut, flattening of the lens. Right, and here you see accommodation. Relaxed ciliary muscles. You see how the muscle kind of bulges a little bit forward, and we have the bulging up of the lens, and we focus more nearer, okay? I'm sorry. This is the distant ones, and they really didn't draw this very well because you can't see the difference of that it's slack. So you have your relaxed muscle, and this should be tightened, okay? And this is showing the tightened muscle. This almost looks exactly the same to me, but the muscle moves forward, and we have a slackening on the suspensory ligaments. And here you see you're focusing on your near object. So to have that accommodation, to see our near vision, we're going to have three major changes taking places. We're going to increase the curvature of the lens. We're going to constrict our pupil, meiosis, okay? My cat is getting back here, trying to knock everything off the table. And we're going to have convergence of our two eyes, which you can see if you watch my finger as it comes closer, you're going to see my eyes kind of focus in closer than when I go out, and that's convergence, the focal point kind of narrowing almost towards your nose. The other disorder with the vision that you want to be concerned about is knowing about presbyopia. So we have myopia, nearsightedness, hyperopia, farsightedness, and then what happens around age 45 to 55 for most of us is presbyopia. Presbyopia happens as the lens loses its elasticity and ability to accommodate for near vision. And that's why you start needing reading glasses to help you focus on your near vision, as that accommodation doesn't work quite as well as it used to, okay? When our pupils constrict, as part of this process, number 2 here, it prevents divergent rays from entering, which would cause some blurring of our vision, okay? So the pupil constricts in the brighter light in order to protect the retina from too much extraneous stimulation. All right, so when our pupil constricts, we talk about the near reflex. That's the pupil that constricts with accommodation. We also have the photopupillary reflex, where the pupil constricts in bright light. So you flash light into your eyes, your pupil constricts. We also have what we did in lab that one day of the consensual reflex, which is where if I flash light in one eye, it doesn't just constrict this eye, it also, with that optic chiasm and the communication of the nerves across each other, will cause the opposite eye to constrict at the same time, all right? Convergence, which I was just kind of showing you, and you can practice at home on somebody, one of your loved ones by moving your finger closer and closer in, is you're going to turn the two eyes inward to look at a close object, all right? Convergence requires your extraocular muscles to work and function similarly, okay? And so your pupils will get a little bit smaller. Convergence, accommodation, and meiosis is the triad, all right? The near vision triad. The closer the object, the greater convergence we need to maintain vision. Now, 20/20 vision. What percentage of us, we all know, most of us probably wear glasses, contact lenses, had LASIK surgery or something. And I thought I'd look up the statistics because I had heard a while ago that greater than 50% of the adults have some degree of visual assistance need. And I see from the University of Iowa, it says only 35% of all adults have 20/20 vision without glasses or contact lenses or corrective surgery. And even with all those things available, we can only get 75% of us up to 20/20 vision. 25% of people will never get there. They're unable to fully correct to 20/20. So, and usually they have a lot of damage like diabetes and hypertension damage and such things to the eye, or, you know, some congenital stuff that causes that to not be able to approach 20/20 vision. Some of the other eye defects we get is strabismus. Okay, strabismus is part of not being able to align our eyes, or sometimes what's colloquially known as a lazy eye. And so one eye has to work a little bit harder, might get there to focus. All right. Now let's talk a little bit about our rods and our cones. So rods will respond to any particulate light matter, also known as a photon, regardless of how much energy is contained. Whereas cones have to have bright light, and they have very characteristic ranges of sensitivity that are in each sort of primary color range. So our rods have a pigment called rhodopsin found in the rods, and it's super sensitive to any light. So even in dim light, it's going to cause the retinal portion of this to change shape and leave what's called the opsin molecule that's bleached out. Separation of opsin from retinal, the two parts of the rhodopsin, will cause an action potential in the rod cells that then gets transferred through those bipolar cells into the ganglion cells that then form the optic nerve. The cones only perceive in a combination of signals from their color type. All right. So the cone pigments are less sensitive than the rods that contain rhodopsin. They need brighter light to break down the pigments in them. And they each respond to a characteristic sort of range. So we say the blue cones are stimulated by short wavelengths. The green cones are stimulated by medium wavelengths, and the red cones are stimulated by long wavelengths. So looking at this, we know that this is the deepest layer of the retina, of the neuronal layer tunic of the inner eye. All right. And so the photoreceptor will get stimulated by the light, and it's then going to transmit the information to the bipolar neurons. Cones to one bipolar neuron, rods, many rods to one bipolar neuron. And these in turn send their messages to the ganglion neurons. This is what forms cranial nerve number 2. So looking at this, we see the rods get stimulated right around 500 nanometers. The short wavelength, 420, is the S cones, or the blue range. The green range is the medium wavelengths, around 530, and the red cones, or the L cones, at about 560. Aren't you glad you get to use your resources for this test instead of having to memorize this stuff? Because this is really kind of useless memorization unless you're doing it all the time, in my opinion. All right, so we had our rod and our cone, one cone to one bipolar, many rods to one bipolar, and then the bipolars transmit their message to the ganglion cells. The ganglion cells will relay these messages, and they also relay, along with it, non-image light for information to help set our biological clock, okay? The neural pathway of vision will be that these fibers from the rods and cones go to those bipolar cells, to the ganglion cells, and then that optic nerve, cranial nerve number 2, will travel, some to each side, but some will cross over at the chiasm, and they'll go from the optic nerves all the way back through the optic tracts to the occipital lobes in what's called the optic radiations. All right. So I'm skipping over disorders of the ears because we'll talk about it when we do the ears. So we'll talk a little bit about refraction disorders, some disorders of the retina, and some disorders of the vision pathway. So visual acuity, 20/20 vision on the Snellen chart, the chart with the big E. This means that you can read at 20 feet what most every human being can read at 20 feet. When you are 20, 40, 50, 2200, getting progressively bigger, this means at 20 feet, you can only read what a normal person could read at 200 feet away from the chart. So your visual acuity is poorer the larger that number gets. That's called myopia, nearsightedness. Hypermetropia or hypertropia would be like 20/15, meaning that you can read at 20 feet what a normal person, you can read at 15 feet what a normal person could read at 20 feet, okay? So your visual acuity is a little bit better, all right? Now, 20/20 is normal. 2200 is where we put the cutoff of being legally blind. But of course, you know, you can refract and get some of these people to 20/20 or awfully close to it with refraction, spectacles, contact lenses, or even LASIK surgery, or, you know, some of the other laser eye surgeries. Astigmatism is something different. Astigmatism happens when the cornea is not symmetric. It becomes, instead of rounded over the lens, it's more football shaped, a little bit curved. And so people with astigmatism have kind of like that dispersion of the light, a little bit of diffraction in the eye. And you can have astigmatism with normal vision, along with astigmatism. You can have myopia with astigmatism, and you can have hyperopia with astigmatism. And astigmatism gives this little blurriness, okay, around the letters. Photons will stimulate both rods and cones, okay? If, when light comes in the eye, it is completely absorbed by all the photons. If all the photons are absorbed, then you see the color black, all right? If they're all reflected, you see white. But when we only absorb color in a particular range, you see the spectrum of that color. So if everything is being absorbed within the blue S cone range, you'll see blue. In the green cone range, 530 nanometers, you'll see green. 560 nanometers in the red cone range, you'll see red. And then you'll get various combinations of colors depending on what is stimulated more or less, all right? Yellow is going to be a combination of highly stimulated green cones with a little bit of stimulation from the red cones, no participation from the blue cones, for instance. All right, so color blindness. Color blindness is when one or more of the cones are not functioning properly. So you can have a missing cone, like you're completely born without having any red cones, or you can have all the cones present, but maybe they're not functioning properly. Maybe they're not making the pigment that absorbs the light. The most common color blindness is red-green blindness, and it tends to be genetically linked and more common in males. 10% of some males are colorblind. Okay, only less than 1% of females are colorblind. So ladies, it's true, he really can't match his socks, all right? Genes encoding the L pigments, okay, remember the L pigments are the red pigments, are linked to the X chromosome. This is why. So looking at this is like one of our basic colorblind tests. If you can't see the number 12 buried in here, you're probably lacking that L pigment cone. All right. That's a red color blindness right there. Let's talk a little bit about glaucoma. This has to do with the circulation of the aqueous humor in the eye. Remember, and why we go over glaucoma is it's so common and it's one of the leading cause, if not the leading cause of blindness. All right, so, whoops. Over the ciliary body, we have ciliary epithelium that makes the aqueous humor. The aqueous humor then circulates from the posterior chamber to the anterior chamber. And if this trabecular meshwork gets kind of clogged for some reason, we're not quite sure on everything that leads into the pathology, but it stops reabsorbing well. We get buildup of fluid, and that increases the pressure. Increased pressure in the eye causes damage to the optic nerve. That is open angle glaucoma. This tends to be a slow process, and we treat it. We give you drugs and such to take care of that, some eye drops. Closed angle glaucoma, you see here that the lens starts pushing forward and pushing this part of the ciliary body and iris up against that trabecular meshwork and the canalus phlegm. This happens acutely, it happens fast, and it's considered emergent. It's a medical emergency. We have to treat it right away. All right. And it happens, of course, precipitously, right? Typically, you start getting blurry vision and such with closed open angle glaucoma. Here, you get like a really bad headache and some eye pain is usually the primary symptoms, along with some visual symptoms. Cataracts. Cataracts are really, really common. Cataracts are when all these crystalline proteins of the lens and that are supposed to be nice and transparent to let the light through. When these cells start losing their transparency and get cloudy, typically in older people, there are some congenital and youthful cataracts, but not real common. The cataract gets foggy and clouded, and so does your vision. All right. So what we do is we end up, you can kind of slowly treat it with some refractive things at first, but ultimately the fix is called an intraocular lens implant. We take out the old natural born lens and we put in a fake lens, okay? A big surgical procedure, but it's done at outpatient clinics, and it's kind of a miracle. You can also accommodate for some refractive problems. It's like Lazarus in the Bible, people who, were dead suddenly awaken in terms of their vision. People are pretty happy with their cataract surgery. Macular degeneration. Macular degeneration is terrible, okay? And usually it's caused by, you're getting, older, and in dry macular degeneration, we get a deterioration of that visual center, the fovea of the macula, the real central part. So the center of your vision degenerates. The harder to treat part is what's called wet macular degeneration. And that's where we get blood vessels growing over the center of our vision. Remember how when we looked at that fundoscopy in the center of the fovea, there was no blood vessels? When blood vessels start growing into the fovea, you start losing your central vision. And this is what happens when you hear about all the elderly having to go in for their eye shots. We put injections into the actual retinal area, some anti-angiogenesis drugs, things that make the blood vessels in the center of vision deteriorate. Now, helping to prevent that is why people take those AREDS vitamin formula, okay? It helps somehow in preventing or reducing a little bit the disease progression of macular degeneration. All right, so this is the little test you can do at home for your blind spot. You can print out this one slide if you want. And while you look at the little plus sign, you can move the picture closer and closer. and try to see the distance at which the dot disappears from your sight. And this is where you can detect your blind spot. Okay. And then you can reverse it and move it away and, see as the blind spot comes back as well. So we have this, you can kind of print it out at home and play around with it. It's kind of fun. All right, we'll look at otoscope later on, talk about this. So let's answer a few practice questions. Why are the rods best suited for night vision? Anybody want to pipe in? Maybe you all have lost interest and signed off. The reason is because our rods have a lower threshold for activation, right? They are activated in dim light. Try and move forward. Whoops. Okay, so that's the area. That's the answer. What processes have to take place for the lens of your eyes to become rounder and focus on nearby objects? So, do the ciliary muscles react and the suspensory ligaments pull at the lens tighter? Remember, for the lens to round up, the zonular fibers have to be loose. Okay, so it's going to be C. The ciliary muscles will contract, they kind of move forward, and that reduces the tension on those zonular fibers. I know it's super counterintuitive. That's why we go over it so much. It's really, really highly tested. Okay. All right. We know what is the optic? Why is the optic disc a blind spot? You can see Regina. Regina, your sound's off, but I can see on your lips, there's no photoreceptors. No photoreceptors, no rods or cones.
Come on, give me something. Come.
Okay. Let's trace the circulation from the aqueous humor from the site of production to the site where it's reabsorbed. So where is it produced? It's made at the ciliary body, the epithelium. So we know we're going to have to begin with 3, and then we reabsorb it in the trabecular meshwork in the canalus phlegm. So we start with 3, we end with 4. The only one that does that is going to be D. Okay. Made in the ciliary body, circulates around the posterior chamber, goes through the iris pupil, and comes up through the anterior chamber and leaves at the canalus phlegm. All right. So there we have it. We're a little bit over time, are we? 7:17? No, we were going to go to 8 o'clock, but we can leave early here, right? Since we're at home. Cool. Carrie to everybody. Carrie's got all the answers here too. All right. You guys feel good for tonight?
Yeah.
Y'all can have a good Thanksgiving without me. You see, oh my God, my throat is so sore. I'm sorry if I'm a little off my game tonight, but I hope the vision makes sense. And we have Wednesday off. So the next time we see each other will be next Monday. We have nothing due right now, but remember, you have the option for those of you who wanted to get 5 possible bonus points that you can work on that special senses lab and hand it in. We had at least 7 or 8 people who were straight scale getting A's. I'm really happy where everyone is. So I think we're going into this last exam pretty good, but remember, neuro's kind of hard. So we'll do lots of practice questions. I'll put practice questions over on the practice part of there's some already up there on that practice section. And since I can't seem to load anything more on Blackboard, if I look through there and I find a deficiency of anything, I will email stuff to you. But make sure to at least take a few days at Thanksgiving off to have gratitude, spend time with your families, and relax a little bit so that your brain is detoxified and you can come back in and make that last, 10-day push that we need to get done with class. Okay. You do better if you take a little bit of time off. Okay, so I'll be like Barbara Walters and say, take some time off to enjoy the view, right?
Thank you, Dr. Ara. Thank you so much.
Okay.
When is the PBL going to be posted?
I'm going to post it on the Sunday night after Thanksgiving because I don't want any of you working on it and being a busy little bee over Thanksgiving. Okay. And you
Okay.
have like 10 whole days to be able to work on it. All right.
Okay, sounds good.
All right. And
No
Kiara,
class on Wednesday.
we're so glad you came through and are feeling better. Is Shakira here tonight? Because I know Shakira wasn't feeling super great either, but I don't see her on here. So I'm going to have to check in with her and make sure we're not losing our little 18-year-old wonder kid here. We all got to put her on, okay? So everybody
Okay.
No class on Wednesday.
Huh?
No, Regina,
class
no class
Wednesday.
on
You guys have Wednesday off.
Thank
Right.
you.
Thank goodness.
Take care.
Thank you.
All right.
Thank
Bye.
you.
Happy holiday,
Bye bye.
everybody.
Have a nice
you.
holiday.
Thank you. I will.
Bye.
See you Monday.
Okay, bye-bye. Bye.
Bye.
Yeah.
And I'll post this too, by the way.
Okay, thank you. Send me word to your mom, okay?
I will. I should have taken my computer in so you guys can meet her.
Yeah, let me see. Let me see her.
Yeah, I think she actually went to bed early because I hear no noise out there, so I can't right now. Besides,
Okay.
my computer is kind of hardwired in right here. I'm not on my laptop. I'm on my desktop. So one of these days, I usually bring in a recorded message from her, and she wishes you all well for your finals. So I'll do that Monday.
Yeah, thank you so much.
Okay.
Thank you.
Bye,
Bye-bye.
y'all.